initial setup. There it goes. Hello, everyone. This is a very, I should say, a kind of an introduction to the project which I have decided to do for the final presentation. This is how I organized it. It's going to mainly talk about the schizophrenia and using some, I should say, machine learning algorithm for, I should say, feature selection and also enhancements for the diagnosis of the schizophrenia based on two modalities, which is extracted from both MRI scans and also fMRI scans. But the nature of that study was somehow that, for example, those modalities and data which is collected from those experiments requires a substantial amount of the study in order to understand what do they mean. Then in order to just make that, I should say, presentation more meaningful and comprehensive, I decided to have some, I should say, some elementary, I should say, introduction to what is schizophrenia. And then I'm going to talk about the brain connectivity and networks of the brain. Then I'm just going to say how the data are collected and how it's going to be optimally, I should say, combined for enhancements for the diagnosis of the schizophrenia. The first thing is that, okay. The first slide is that it's a very quick introduction about the schizophrenia. What is the schizophrenia and what kind of, I should say, symptoms does it have, which is going to be, I should say, embedded inside the prediction of enhancements of those features which result in the prediction. One of the most symptoms which you mostly have heard is going to be hallucinations. It's going to be also delusions. And also, it's going to be catatonia, which is the kind of symptoms that the people who are schizophrenic, they have some kind of postures and movements, and they stay in that position for a very, very long time. I have a very, I should say, very quick video that it shows that the schizophrenic patients, which showing some kind of that kind of symptoms.
Like a warm candlestick, the body of the patient can be contorted into almost any posture. The patient may maintain the posture for several minutes.
Oh, you did not see that video, huh?
I don't think you want to show the video, if that's too graphic.
Okay. Let me then go away from... It was a very cool, I should say, 30 seconds showing that schizophrenic patients, they contort their body postures and they stay in that position for a very, very long time. Okay, and the other thing is that that's a very quick, I should say, showing that what is the mechanism behind a person who develops the schizophrenic symptoms. Normally, the schizophrenia shows up in the patients within the age of 16 until the utmost of, I should say, 35 years of age. And there is, I should say, a very minor possibility that someone below the age of 16 or above the age of 40, they show that they're schizophrenic. There are two theories behind the development, I should say, of this schizophrenia. There are two kind of pathways and hypotheses which they result to the appearance of those symptoms. One of them is dopamine hypothesis, which is a kind of a neurotransmitters inside the brain, synthesize the brain, and also glutamate hypothesis, which also is another neurotransmitters synthesized inside the neuron of the brain, and I should say some kind of dysregulation of those pathways results in schizophrenia. For example, as a very last slide on, I should say, the pathway dysregulation which causes schizophrenia is that slide which shows that, for example, in the dopamine hypothesis, we have, I should say, inside the brain, we have 5 distinct pathways which they synthesize, the neurons in that pathways, they synthesize dopamine, which is also included, I should say, implicated in Parkinson and also at the same time with schizophrenia. Out of those 5 pathways, the most important one is kind of mesolimbic pathways, which, as you will see on this, I should say, on this slide, which is going to be on the left, it starts somewhere from, I should say, ventral, the telgora, I should say, area. It starts from here and goes all the way to the nucleus accumbens of the brain. And all those neurons inside that area, which is, I should say, the midbrain of, I should say, the patients, they are involved in synthesizing dopamine. And any hyperactivity and disturbed, I should say, activation in that pathways only, it may result in the symptoms of the schizophrenia. Okay, and now the most important thing of my project is talk about, I should say, brain connectivity and networks of the brain, which essentially these kind of data sets are extracted from the fMRI scans, and MRI scans try to, I should say, represent the symptoms of schizophrenia. But when we talk about, I should say, the brain connectivity, we are talking about, for example, how two, I should say, regions. Normally, they call that, in terms of the MRI or fMRI, two voxels of the brain. For example, if you imagine a brain as a kind of a three-dimensional, I should say, squares, if you divide that based on a single unit, which is going to be the whole brain, it can be divided into 100,000s, like small units. Those small units, they are called voxels. And you're looking to see that, for example, one voxel, which is located on the forebrain of the, for example, of the skull in the brain, how is that going to be connected? Essentially be correlated with another distant region of the brain, for example, into, for example, the midbrain, or even to, let's say, for example, cerebellar part of the brain, which is the lower part of the brain. And that's essentially the object of talking about the neural connectivity. And normally there are two views, I should say, on the brain connectivity. And one of them is that they're talking about the, let me just go to the next slide. They're talking about, I should say, the localization of, there is one perspective, and they call that's gonna be the localization of the neural connectivity. Essentially, they mean that, for example, any connections of your brain, any connectivity of your brain, it localizes with one mental mood. Or vice versa, any mental mood which you have, for example, if you feel depression, it can be localized into one single distinct region of your brain. That's what they mean by the localization of the functions of your brain. Also, for example, there are some other views. They call that, for example, the functional globalist, which they say that, for example, the whole region of the brain, they're going to be involved in, for example, in your mood. For example, there has been an experiment. They showed that, for example, a very small cortical lesion on the brain of the rat, it resulted on the global change of the behavior of the rat. Then these two perspectives needs to be, I should say, taken into consideration. And also, one other thing which I just want to show that from my presentation, for the research which I'm just going to do, I'm just going to more focus on the functional localization, which I mean that, for example, any mental mood, any mental disorders, you can localize that specifically on one part of the region of the brain, and then as a result, when you just want to get all, I should say, all the data from the correlation of the functional connectivity, you need to, I should say, they call that functional integration. Then you have to integrate over all those, I should say, localized, I should say, modality to represent, I should say, the human behavior and mood. Then for the rest of the presentation, I stick to this first assumption, to localize the function with behavior. Okay, and as I talked about, for example, the functional connectivity, it means that you just try to find the correlation coefficients between different parts of the brain. And in that sense, you can essentially infer two different concepts. For example, the first one you can just have is that, for example, if you have a correlation in the functional network of two regions of the brain, the first important thing which you can deduce and infer from that is that you can see, for example, how bilaterally, as for example, even you can just show that in a graphical model, how one part of the brain, one voxel impacts the other voxel, and vice versa. Or at the same time, for example, you have for the visual cortex, for example, when two parts of the brain get the same input from one other part of the brain, for example, visual cortex, then in that case, you see that when you have a stimuli which has the same inputs for the other two parts of the brain, you see that they are not directly connected, but you can define a correlation coefficient between these two parts of the brain, which they have the same inputs from one specific part of the brain. And also, we are just going to get close to some kind of computation of those kind of brain connection and network connectivity of the brain. But before that, I think I just missed one very interesting, I should say, parts of the neural connectivity, which is kind of an example. If you see that on this slide, I just got that from the Wikipedia. I forgot to just put the reference for it. This is from the Wikipedia. And if you see that, it's a kind of a grand scale of neural connectivity in the default mode of the brain, which it means that, for example, in situations when you are, for example, daydreaming, you are not focusing on an external stimuli, the brain has some kind of a pattern as a default mode that the neurons in that brain, they are just connected and they are just kind of firing pathways between those two kind of the regions, which is, I should say, this is, for example, you will see that these parts of the brain, they are called that default mode networks of your brain, which there has been some kind of, I should say, if I just want to digress, has been implication of this network with the patients who develop the Alzheimer. Okay, and now just go to some kind of computations of my research, is that you have then two kind of connectivity, the functional connectivity and the effective connectivity. Effective connectivity was a kind of, for example, the bilateral interaction of two parts of the brain. In that sense, for the functional connectivity, there are three different methods. The first method is that, for example, you do the seed analysis, or also at the same time, which is a kind of a univariate analysis of the functional connectivity of the brain, and also at the same time, sometimes you have some, I should say, drawbacks from the seed analysis, and you have to just adopt a kind of a multivariate, I should say, approach to your, I should say, correlation coefficients of your neural connectivity, which, for example, you will see that you have, for example, PCA method. I'm just going to talk about that a little, and I should say also independent campaign analysis, and at the same time, partial least squares, which sometimes gives a better result compared to the previous result. And also for effective connectivity, there are some other, I should say, methods like structural equation modeling, granular causality, and BiasNet. Okay, the result of my, I should say, one part of the data which I have extracted from my analysis is derived from that method. This method shows that, for example, you have two regions of the brain, region 1 and region 2, and you just try to, for example, you present the patients, schizophrenic patients, with a stimulus. And in that sense, you try, it shows that in the fMRI technology, there's a terminology, they call that hemodynamic response. It means that, for example, the law of the electricity changes the oxygen in the neurons with respect to the changes of the activity, it's monitored with respect to the time. And you will see that you have some kind of, not Gaussian, you see some kind of a curves, which shows that, for example, for subject 1, patient number 1, after, I should say, data stimuli, you have data, I should say, peaks generated. You come to use, I should say, a collection of the regressors or estimators as a general linearized model to find, I should say, some parameters in a parametric approach, in your statistical modeling, to find, I should say, some parameters of each hemodynamic, response, each peak. And in that sense, you have some parameters, and those ones also, for example, can be a kind of a data to be processed further to give the connections and, I should say, correlations. For example, with these ones, you find the correlation coefficients R. Then you use Fisher transformation to transform them into the Z to make them as a kind of a Gaussian distributed, I should say, correlation coefficients. And also, sometimes when you have some kind of control, for example, in the previous model, if the stimulus is presented to the schizophrenic patients, because there are some kind of a time series analysis, if, for example, the timing sequences between each stimulus is going to be so short, there's a kind of an overlapping between each, I should say, peak on the hemodynamic graph. There are two options to do that. The first one is that, for example, you can just use a ridge regression when you just want to use, I should say, your regression models to estimate those parameters. Or at the second time, you can just use some kind of multivariate decompositions, which is going to be the second type of my data for the SPM is extracted from these methods. For example, for the principal component analysis, you have, for example, for each cortex, you detect, for example, the intensity of the fMRI data sets. And you know that in the matrix, you have a two-dimensional matrix. The rows is going to be your time points. The column is going to be the voxel intensities. And you just use, I should say, the PCA technique to find the most, non-related, orthogonal, but most explaining the variations in each difference, difference of your voxel intensities. But it has two drawbacks. The first one is that it's only sensitive to, I should say, it's a very important thing. The PCA is very sensitive to those voxel distribution, the voxel intensities, which they are statistically distributed by Gaussian. But lots of, I should say, voxel intensities, they are not Gaussian distributed. Then when you have the PC analysis on them, you have those intensity of the voxels merged together in one direction, and you lose track of them. Then in that case, you have to just, for example, you can just use a different method, which is essentially that thing which I have done on the data set, which is going to be independent component analysis of the functional connectivity, which you know that, for example, it's a problem of the blind source separation. It's from the literature. For example, imagine that you are in a cocktail party, there is music from different sources, and you record all those musics, but at the same time, you just want to do some kind of analysis in your data to extract each source of the musics. For example, you have the separate, I should say, sources, not combined together. And that's going to be done by this method. It's a kind of a probabilistic ICA, because at the same time you have to just introduce an error term, which is, I should say, a non-Gaussian, a kind of a Gaussian, I should say, error term at the end. And also there is one very quick drawback of that method also. The first drawback is that when you use, I should say, probabilistic independent component analysis on your brain connection, that method is stochastic. Then when you have, for example, the same sets of data sets from the fMRI and MRI, which they represent, I should say, the voxel intensities, if you run the same algorithm on the same data sets every time, you get a different results as a result of, I should say, of using your probabilistic independent component analysis. That's one drawback. And then the thing which I took this one from the papers, which I'm just working with the data sets on that, is that they talk about for extracting the second type of the data sets. Because in my project, I have two sources of the data. The first one is a functional neural connectivity, which is essentially the one which I showed that to you. They call that beta coefficients, which is a kind of a general linearized method on neural connectivity. You get the parameters, then you have the correlation coefficients. You use a Fisher transform to get, I should say, your raw data. But for the second source of the data, you are talking about the source-based morphometry. What is that? How you can just get that from, I should say, brain connection and brain networks. Essentially, the thing which you do is that you get your, I should say, a time series of your, let's say, MRI, structural MRI data sets. And you have analyzed that based on a voxel. Then you try to find, I should say, as on time series, try to find the correlation between each voxel with, for example, another time steps in, for example, taking those MRI scans. In that case, you call that voxel-based morphometry. You try to find a kind of an indication of the structure of the brain of the subject which is under, let's say, the MRI experiment. But the problem of the voxel-based morphometry is that when you try to do some kind of statistical test on single voxel, you do not find, it's not a kind of a generative method, because you do not find, let's say, any information and any relation with the other neighboring voxels of the brain which impacts that voxel. Then as a result, you try to minimize that error by using, I should say, a kind of a source-based morphometry, the second type of the data sets, which essentially is the statistical analysis on each voxels, but using independent component analysis to find, I should say, those data sets, those kind of, I should say, non-Gaussian, because by ICA you move towards non-Gaussianity of your data sets. That explains, I should say, the most variation. And by doing that, you will find that, for example, if you use that technique, for example, that's the structure, for example, with voxel-based morphometry, you pre-process your images, do a statistical analysis, and you do a statistical map of your brain. But with the source-based morphometry, between these two steps, you run, I should say, your independent component analysis. Then you run your statistical analysis based on non-Gaussian, I should say, data set vectors which you have derived from that step. And based on that, I should say, technique, you will discover that, for example, for a person who has been diagnosed with schizophrenia, you will see 5 different regions of the brain, which it shows that those 5 regions of the brain, they are highly correlated, highly localized, especially localized, to show that the person with schizophrenic, they have, I should say, some kind of dysregulation in those parts. For example, from bilateral temporal lobe, thalamus, basal ganglia, parietal lobe, and frontotemporal regions. But if you do the same experiment with the same patients, not using the ICA on your fMRI datasets, you will see that the only region of the brain, which is a kind of a localization of the schizophrenia, is only the thalamus. Then using the ICA gives more information of, let's say, about the regions of the brain which are localized for the schizophrenia. Okay, let me... these are essentially the method which is from this paper. On the top right of that slide, you will see the paper which I'm just working on it based on the techniques they have used. But to use the ICA, they have used, I should say, they have used, instead of using the regular machine learning for the ICA, because of the huge data sets, they have used a kind of a neural network to get and used, I should say, they're trying in that neural network to minimize the mutual information as an output of the neural nets. And okay, that's essentially all the I should say, the knowledge which it was required for that thing. I'm just showing you, for example, some kind of data specification, how the data looks like. For example, this is kind of in a slide which I have, I should say, screen, I should say, shot the data on the R. You will see that, for example, for the functional neural connectivity, the data sets contains, I should say, for the training data sets which I have, it contains of 86 observation, which means that 86 patients, and it has 379 variables. By the 379 variables, I mean the functional neural connectivity. All those data, they are somewhere between -1 to 1, which they are essentially the correlation coefficients of the Fisher transformation on your, I should say, linear model, which you had on your beta coefficients. And also, for the SVM, source-based morphometry, the data set looks like that. You have, I should say, in this case, again, you have 86 patients, schizophrenic patients, but you have, I should say, 33 features. I mean, 33 outputs from your neural nets, which I should say computed, I should say, your functional connectivity of the voxels, filtering that, if I can, if filtering is the right word, I mean using the ICA on those the features, and you have 3 sets, which it shows that the kind of structure indications of your brain of the schizophrenic patients. At this stage, I'm trying to optimally connect these 2 data sets together, because the first data sets, F and C, represents the correlation coefficients after Fisher transformation of all parts of the brain in fMRI data sets. But this one is from the MRI, I should say, scans, which represents, I should say, some kind of a structure of the brain. Initially, I just combined them in a very, very simple formats of the R. I ran, I should say, random forest to extract the features. I got the features which is relevant for the schizophrenic patients, but I'm still working on it to find the best algorithm for optimally combine them if there is any algorithm available. And also, I'm going to talk about very quickly, I think I just ran over time, it's 23 minutes I'm talking, very quickly about the expected outcomes and the challenges which I faced for that project. The challenges, first of all, is that I'm just using 2 different modalities of the brain. One coming from the fMRI experiment, the other one coming from the MRI experiment. And I have to optimally combine them together. For example, the right, I should say, correlation coefficient matches exactly, not exactly, if I just do some kind of probabilistic approach to that, with some kind of an uncertainty with, I should say, with the SPM datasets. This is one of the biggest challenges, and the expected outcome is that after optimally combine them together, I just want to extract the features which they can be more relevant in terms of, I should say, schizophrenia symptoms, I should say, diagnosis. It's like a kind of regions of the brain which localize when you just want to just make that diagnosis. And that's pretty much. If you're interested with the slides, we can just, this is my, I should say, GitHub, I should say, address. I'm just going to post that on my GitHub. And that was all about my presentation. Thank you. It was 25 minutes. Thanks. Any questions or comments?
Certainly very energetic. That's very good. ICA certainly is the one that will use. I just want to be for the class to know that ICA independent component analysis is one they use a lot. There's a reason for that. In the summer, in August, there was the academic, the grandson of the inventor of the EEG was teaching a workshop on campus.
Here at York.
Yeah, I couldn't send that to everybody because very frequently it's $1,000 for 2.5 days. But then he talked about a lot of the ICA. So the ICA seems to be very, very effective for analyzing brain data.
That's right.
Yeah. That's one to comment.
Thanks a lot. No questions?